Welcome to Podcast Learn and Fun. Let's start today's journey of learning. Imagine you're lactose intolerant, but you really want a milkshake. Well, thanks to enzymes, you can actually have your shake and drink it too. That's because we've figured out how to use a specific enzyme called lactase to break down that pesky lactose sugar in milk, making it digestible even for those who usually can't handle dairy.
Pretty neat, huh?
This is just one tiny example of the amazing power of enzymes, those microscopic protein machines that are essential for practically every process in living things.
Yeah. You see, what makes enzymes so incredible is their efficiency. They act like catalysts, speeding up chemical reactions in our bodies by a factor of thousands or even millions.
Wow, that's incredible.
And get this, they're not used up in the process.
Really?
They're reusable, like tiny, tireless workers on a cellular assembly line. They latch onto specific molecules called substrates, like a lock and key. They help transform them into new products.
This lock and key idea is fascinating, but I'm having trouble picturing it at the molecular level. How does this interaction actually play out?
It's like a dynamic dance, really. The enzyme has a special pocket called the active site, and when the substrate molecule comes along, it fits perfectly into that pocket. But it's not just a rigid fit. Sometimes the enzyme actually adjusts its shape slightly to get an even snugger grip on the substrate, like a glove molding to your hand. We call this induced fit. And once that perfect embrace is achieved, the enzyme helps lower the energy needed for the reaction to happen, making it occur much faster.
I see. So it's not a static lock and key. It's more like a dynamic interaction where the enzyme can actually change its shape to optimize the reaction. That's pretty cool.
Exactly. It's a very elegant process.
I'm trying to wrap my head around how researchers first figured this intricate dance out. What kind of experiments did they conduct?
Early pioneers in enzymology, like Haldane and Michaelis-Menten, had to rely on clever deductions and mathematical models to understand how enzymes worked. They painstakingly measured reaction rates under different conditions, like varying the amount of substrate. All this to figure out the relationship between enzyme speed and substrate concentration. Their work laid the foundation for a whole field of study called enzyme kinetics, which uses math to describe how enzymes behave.
It's mind-boggling that we can actually witness these molecular transformations happening. How is that even possible?
Thanks to some incredibly powerful tools, we've gone from theoretical models to actually seeing enzymes in action.
That's amazing.
X-ray crystallography and NMR spectroscopy allow us to visualize the three-dimensional structure of enzymes.
Wow.
Almost like taking a snapshot of them frozen in time. We can actually see how their shapes change as they interact with substrates and catalyze reactions.
Knowing how vital enzymes are, you might be wondering what could possibly disrupt these intricate processes.
That's a great question.
Well, that's where inhibitors come in, right? They're like the villains in the enzyme world.
Exactly. Inhibitors are molecules that can mess with an enzyme's ability to do its job. Some inhibitors, called competitive inhibitors, are like bar hoppers trying to steal each other's stools. They compete directly with the substrate for that coveted spot on the enzyme's active site.
So they're basically vying for the enzyme's attention, trying to block the substrate from binding.
Exactly. Sometimes they win, sometimes they lose. It depends on who's more persistent and who has a stronger affinity for the enzyme.
And what about those sneakier, non-competitive inhibitors? How do they wreak havoc?
Instead of a direct confrontation, they opt for sabotage. These crafty molecules bind to a different part of the enzyme, away from the active site, but their presence still manages to distort the enzyme's shape, making it harder for the substrate to bind or for the reaction to occur efficiently. It's like throwing a wrench into the enzyme's machinery.
So we've talked about inhibitors messing things up, but aren't there also molecules that can help enzymes do their jobs better? Like a trusty sidekick to the superhero enzyme.
You're absolutely right. Some enzymes need a little help from their friends to function optimally. These helpers can be metal ions, called cofactors, or organic molecules, known as coenzymes. They're often essential for the enzyme to work properly, kind of like how a mechanic needs the right tools to fix a car.
It's amazing how this all works together in such a coordinated way. It's like an elaborate symphony playing out at the molecular level.
And this intricate symphony isn't just about individual reactions. It's about orchestrating entire chains of events within the cell. We call these interconnected series of reactions metabolic pathways. Think of it like a complex assembly line, where each enzyme is responsible for a specific step in the manufacturing process. The product of one reaction becomes the substrate for the next. and so on, creating a cascade of transformations that ultimately lead to the creation of essential molecules or the breakdown of waste products.
So enzymes aren't just speed demons. They're also master choreographers, making sure the right products are made in the right amounts at the right time. That level of precision is astounding.
And just like in a choreographed dance, timing is everything in metabolic pathways. If one enzyme is out of sync, it can throw off the entire routine. This can lead to a buildup of toxic substances, a shortage of essential molecules, or even disruption of vital cellular processes. It's like a domino effect, where one mistake can cascade through the entire system.
That sounds like it could have serious consequences for the organism.
It absolutely can. Many diseases are actually linked to enzyme malfunctions. For example, certain types of cancer can arise from mutations in genes that code for enzymes involved in cell growth and division. When these enzymes go rogue, cells can start multiplying uncontrollably, leading to tumor formation.
So understanding how enzymes work is not just about appreciating the beauty of nature's design, it's also crucial for developing new treatments for diseases.
Exactly. Enzyme research has led to breakthroughs in medicine, agriculture, and biotechnology. We can now design drugs that specifically target certain enzymes, manipulate enzymes to produce valuable compounds, and even use enzymes to diagnose and monitor diseases. It's a field with incredible potential for improving human health and well-being.
It's inspiring to think about how something so small can have such a huge impact. Speaking of impact, I'm curious about how scientists actually study these tiny but mighty enzymes in the lab. What kind of techniques do they use to unlock their secrets?
There's a whole toolbox of methods that scientists use to investigate enzymes, each with its own strengths and limitations. One common technique is called an enzyme assay. It's basically like running a controlled experiment to measure how fast an enzyme can catalyze a reaction. By varying factors like substrate concentration, temperature, and pH, we can learn a lot about how an enzyme behaves under different conditions.
So it's like putting the enzyme through its paces to figure out its operating manual. What other tricks do scientists have up their sleeves?
Well, we can also use techniques like gel electrophoresis to separate and identify different enzymes based on their size and electrical charge. It's like a molecular race, with the enzymes zipping through a gel at different speeds, depending on their properties. This allows us to isolate and study specific enzymes from a complex mixture.
That sounds pretty high-tech. Are there even more advanced methods for getting up close and personal with these molecular machines?
Absolutely. We have techniques like X-ray crystallography and NMR spectroscopy, which allow us to actually visualize the three-dimensional structure of enzymes in incredible detail. It's like taking a high-resolution snapshot of the enzyme's architecture, revealing the intricate arrangement of atoms that make it work. This structural information is essential for understanding how enzymes bind to substrates, catalyze reactions, and interact with other molecules.
It's amazing how far technology has come, allowing us to peer into the molecular world with such precision.
It really is remarkable. And as technology continues to advance, who knows what other incredible insights we'll gain into the world of enzymes. It's a field that's constantly pushing the boundaries of our understanding of life itself.
It seems like we've only just scratched the surface of this amazing world of enzymes.
We have, but what we've covered already highlights just how essential enzymes are to life as we know it. Without them, the chemical reactions in our bodies would happen far too slowly to sustain us. It'd be like trying to run a marathon at a snail's pace.
And it's not just about keeping us alive, right? Enzymes have a hand in so many different processes, from breaking down food to fighting off infections. It's incredible to think about the sheer scope of their influence.
Absolutely. They're involved in everything from DNA replication to building complex molecules to even helping fireflies glow. Their diversity and versatility are truly remarkable.
Speaking of diversity, I'm curious about the practical applications of all this enzyme knowledge. How is our understanding of these tiny wonders being used to solve real-world problems?
Well, one area where enzymes are making a huge impact is in medicine. For instance, we've been able to develop enzyme inhibitors as drugs.
Oh.
These inhibitors work by blocking the activity of specific enzymes that contribute to disease processes. Think of it like targeting a faulty gear in a complex machine to prevent it from malfunctioning.
So it's like using our knowledge of enzymes to outsmart disease at the molecular level.
Precisely.
Are there other areas where enzymes are proving to be valuable allies?
Absolutely. Enzymes are also making waves in biotechnology. They're being used in everything from food production to biofuel synthesis to environmental remediation. For instance, in the food industry, enzymes are used to improve the texture, flavor, and shelf life of many products.
Oh, wow.
Have you ever noticed that milk is easier to digest now than it was years ago? That's because the enzyme lactase is often added to break down lactose, the sugar in milk that many people have trouble digesting.
So enzymes are helping us enjoy our food more and stay healthier at the same time. I've also heard about enzymes being used to develop more sustainable energy sources.
That's right. Scientists are exploring the use of enzymes to break down plant material, like cellulose and hemicellulose, into sugars that can then be fermented to produce ethanol and other biofuels. This could potentially reduce our reliance on fossil fuels and pave the way for a greener future.
It's amazing how something so tiny can have such a huge impact on so many different aspects of our lives.
It really speaks to the power of scientific inquiry and the importance of understanding the fundamental building blocks of life. As we delve deeper into the world of enzymes, who knows what other incredible discoveries await us? The possibilities are truly limitless.
Well, I think it's safe to say that enzymes are pretty amazing.
I couldn't agree more. They are the unsung heroes of the molecular world, working tirelessly behind the scenes to keep us alive, healthy, and functioning.
Thank you for listening to Podcast Learn and Fun. If you like our videos, please subscribe, click the like button, comment, and share. See you next time.